Welcome to everybody to today's lunchtime seminar for the Department of Psychiatry and CPFT. It's a big pleasure to have Oliver here with us. Oliver is also Professor Howes is a professor of psychiatry, molecular psychiatry, Institute of Psychiatry in London, but also program leader at the MRC London Institute of Medicine and Imperial College. He is also a consultant psychiatrist working hard on people with treatment-resistant schizophrenia, leads one of the most pioneer and interesting groups in of psychiatry about how to start clozapine, and also has done a lot of work on clinical work on treatment-resistant schizophrenia, leading some of the groups worldwide. Can everybody mute themselves, please? Thank you. His research, which is wide and very interesting, as you will see, spans from affective and psychotic disorders. He has one of my top 3 all-time best papers ever, which is, if you can have a time, the Lancet 2014 about the schizophrenia as an integrated socio-developmental and cognitive model. If you have not read that, don't miss it. It's a high-top paper. But today we'll present us all his work on understanding the role of dopamine, especially, and neuroinflammation in the development of psychosis. There's a long range of interesting awards, but perhaps we can discuss that later as well. And we have not convinced him, or we have not managed to convince him to put a slide about where the best ice creams in the world are, which seems to be an expert. But please let me welcome Professor Howes in his talk. Remember that this talk is recorded, so if you have any questions, any comments, and you don't want to be recorded, just turn off your camera. But if there's any comments or any questions, put that in the chat, but we'll get back to after the talk. Thank you very much, Professor Howes, and welcome.
Thank you, Emilio, for that kind invitation. It's a great pleasure to talk to you today about going beyond 60 years of D2 blockade. And I'm going to talk about some of the research that we've been doing and others that will hopefully take us beyond D2 blockers for psychosis. Because let's face it, as Emilio said, I'm a clinician, and antipsychotics, they work well for some patients, but they have lots of issues, and we'll cover some of them today. Just to begin with, I want to give you my acknowledgements. A great many people who've helped with the studies that we've done that I'm going to talk about today, and of course, importantly, the patients and volunteers who've taken part in the studies. I'm also going to show you my disclosures, and as you heard from Emilio, I'm a consultant psychiatrist at the Maudsley, where I see lots of patients, particularly those with treatment resistance or other problems, side effects, etc. But I've also done work with a number of different pharmaceutical companies, because I think we need to engage with them if we're going to get new treatments. So what am I going to cover today? I'm going to talk about the nature of dopamine changes in schizophrenia, but also some new evidence about this in other psychotic disorders, like bipolar disorder. I'm going to talk about changes in people who haven't yet developed psychotic disorders, people at risk of them. And then I'm going to talk about what might underlie them and what this might mean for treatment. And finally, talk a little bit about treatment resistance as well, and some new work that's going to be going on, just starting, including in Cambridge and Peterborough. So, to start with, the nature of dopamine changes. The idea that dopamine has something to do with schizophrenia goes back many years, and it's featured in all the textbooks. This is a cartoon from Stahl's Essential Pharmacology, but all the textbooks pretty much put forward this hypothesis that there is overactivity in the dopamine system in those mesolimbic projections. And the mesolimbic projections, just to remind you, are from the midbrain up to the ventral striatum and to other limbic regions in the brain. Now, this theory's been around for a long time, and it's certainly something that hasn't been disproven yet. I'm going to show you some of the in vivo data that tests it, But I'd also tell you that the textbooks are almost certainly wrong about the mesolimbic localization of this overactivity. And we'll come on to that a little bit in my talk. So how can you test this in patients? Because this theory was really based on findings in rodents and rodent models. Well, you can do that using imaging techniques such as PET and SPECT. So these are molecular imaging techniques that allow you to measure with very high molecular specificity different aspects of the dopamine system. So this cartoon of the dopamine synapse shows that you can measure dopamine receptors postsynaptically, you can measure the transporters, you can measure the proteins that package dopamine into vesicles, and you can even measure the synthesis of dopamine from tyrosine to its precursor dopa, and then its synthesis into dopamine. And that's something that I'm going to talk about quite a lot of, because that's a technique that we've used extensively. We use labeled dopa, which is taken up and converted into dopamine by the enzyme in dopamine neurons called AADC. And it then gets packaged into vesicles ready for release. And so you can measure that process in vivo in patients, and you can also measure the release of dopamine. So this actually allows us to test hypotheses directly in patients and see how they relate to symptoms, which I think is really quite powerful. And people have now done that quite extensively using these techniques in schizophrenia. And I don't have time to go through all the dopamine findings in schizophrenia, but the most marked findings are in this synthesis and release capacity. So using techniques like labeled dopa. And here you see a meta-analysis showing you all the studies. And you can see that by and large, they show large effect size elevations in people with schizophrenia. Summary effect size is 0.8, which is a large effect, highly significant. So overall in schizophrenia, there is this upregulation in the synthesis and release capacity. But the eagle-eyed amongst you will notice that there are one or two studies that don't seem to show a significant difference from controls. And we'll come on to those later. So that's very interesting. It tells us that there is something going on in the dopamine system in schizophrenia. Where is it in the brain? Well, those studies were all in the striatum. And actually you can look at the statistical pattern of statistical changes in the brain in these sorts of scans. And here I'm showing you the data from one of our studies in people with schizophrenia. So this, in yellow, you see areas where there's statistically significant elevation in dopamine synthesis in people with schizophrenia. Just to orient you, the non-imagers amongst you, this is where the ears would be. So this image is looking from the front, this is looking from the top, and this is looking from the side. I have to thank Prince Charles, who very kindly lets me use this picture of his ears to illustrate my talks. So thank you very much. What you can see here is that the largest, most significant areas of elevation are not actually in the ventral striatum, but actually more dorsal, particularly in the caudate. There's something else interesting that is shown here, which is this little blob here, which is in the midbrain. And actually that's something that is present even on some of the early studies in schizophrenia, but was not commented on. But what this actually tells us is that there is overactivity not just in the terminals, the striatal terminals of the dopamine neurons, but also in the midbrain as well. Now, so that fits with the midbrain overactivity model, but the mesolimbic, it doesn't fit with the mesolimbic model because that would suggest the most major overactivity would be in the limbic regions. It actually suggests a mesostriatal model of dopamine overactivity. But do come back to that, or remember that midbrain activity, because we'll come on to that when I show you some further data later. So that's in schizophrenia. But one area that really, one thing that really interested me as a clinician is that, of course, we see lots of psychotic disorders, and the treatment is pretty much always the same. You give them an antipsychotic, a dopamine D2 receptor blocker. And of course, that raises the question, well, they respond to the same treatment. Do they have the same mechanism underlying it? Surprisingly, no one had looked at this until we did a study in people with bipolar disorder. I should say no one has looked at it in psychosis in psychiatry until we did this study. So we did the study in patients with bipolar disorder who were psychotic and compared them to healthy controls, who you see in dark blue on this chart, and patients in schizophrenia in red. And you see on the vertical axis the level of dopamine synthesis capacity. So what you can see is that patients with schizophrenia in red show elevated dopamine synthesis capacity. No surprise there. As I showed you, the meta-analysis has already shown that there's consistent evidence of it. But there's also evidence here that the people with bipolar disorder who show psychotic symptoms show exactly the same elevation in dopamine synthesis capacity. And in fact, it's not significantly different from the patients with schizophrenia. Although I should point out that this study was not designed to test a difference between schizophrenia and bipolar, but actually to test whether the same abnormality was present in bipolar relative to controls, which is what it seems to show. What's more, when you look at the relationship with psychotic symptoms, which is shown here, and they're shown here on the vertical axis, you see that there is a positive correlation with the level of striatal dopamine synthesis capacity that's seen in the patients with bipolar psychosis, who are shown in the black circles, but also in the patients with schizophrenia in the blue triangles. And that relationship seems very similar between the two groups. So the individuals with more dopamine synthesis capacity in the striatum are those that show the most severe psychotic symptoms. So again, it seems to tie this abnormality in the dopamine system to the symptoms that we see in the clinic, and this suggests that actually the relationship is very similar in both schizophrenia and bipolar patients with psychosis. We looked at whether manic symptoms might be influencing this relationship in the bipolar patients, and found that didn't seem to be the case. So, you know, this seems to be specific to the psychotic symptoms, or largely driven by that at least. So that's in bipolar psychosis. What about other psychotic disorders? Well, we went on to do a study with colleagues in Hong Kong, who have a fantastic first episode service. And this is Eric Chen's team, and they were able to recruit and scan first episode patients with a range of different psychotic disorders. And here you can see the results. The dopamine synthesis capacity on the vertical axis again. It's lower here than we've seen in our studies, and that's because of a difference slight difference in their technique that they used, which means they underestimate dopamine synthesis capacity. And then you can see on the horizontal axis the different groups of patients with psychotic disorders, and on the far right, healthy controls. So they were able to recruit and scan patients with delusional disorder, as well as patients with schizophrenia, and then patients with other psychotic disorders. These were predominantly psychosis NOS, sort of brief psychotic episodes that didn't meet full criteria for schizophrenia, although we know from other studies that the majority of them will probably go on to develop schizophrenia, but one or two of them may develop bipolar as well. So what's really interesting here is that you see a significant elevation in the delusional disorder group and in the other psychotic disorder group, as well, not surprisingly, as you see in the schizophrenia group, compared to the healthy controls. So it seems that this change in the dopamine system is present across psychotic disorders, that patients, the biology in patients doesn't follow different, the DSM categorizations that we use, but actually is common across them. So that's very interesting. It tells us that there seems to be this common neurobiology linked to psychosis that we see in patients. But of course, the theory is that this is leading to the onset of psychosis. So how might you study that and test that theory? Well, of course, you can do that by looking at people who've not yet developed the illness. So what we've been doing recently, and others have in other research groups in the world, is looking at people who are at risk of psychotic disorders and looking at the dopamine system in these individuals. Now, we've done some work in people who have prodromal type symptoms, and that's been published. I'm not going to talk about that today in detail, but it essentially shows evidence for an elevation in these same dopamine parameters even before the onset of psychosis, and it was specific to those, or it seems to be specific to those people that go on to then develop the full-blown psychotic illness. So that is consistent with this role of dopamine in the development of psychosis. But those individuals have already developed some subclinical symptoms. So we wanted to look at people even earlier in the development of their psychotic illness. And so we've just done a study in people at high genetic risk for schizophrenia and other psychotic disorders. So these were people that never developed a frank psychotic illness, but have a major genetic risk for it. And that genetic risk is a deletion of DNA at chromosome 22q. This deletion is shown here on the slide, and you can see that it's quite a large deletion, about 4 megabases of DNA, and it includes quite a few genes which are listed across the middle of the slide. One of those genes is COMT, which is a gene that codes for a protein involved in dopamine metabolism. So there's good reasons to think that losing one copy of this gene might affect the dopamine system. Some of these other genes also act on brain neurochemistry, or potentially do, but many of them are also transcription factors which would affect brain development, and we don't fully know what their effects are. But it's quite clear that at least one of the genes in this deletion acts on the dopamine system. Now, having this deletion, which is one of the commonest deletions, big deletions that occur in humans, present in about 1 in 3,000 live births, that increases your risk of schizophrenia massively. So people that carry this have about a 40-fold increased risk of schizophrenia compared to you or I. And over their lifetime, getting on for half of them will develop a schizophreniform disorder, predominantly actually schizophrenia. And actually a good proportion of them develop it by their mid-20s. So it leads to a really high risk of developing schizophrenia and developing it quite early in your life. But what has emerged in the last few years from genetic studies really takes the story to the next level, because people have recognized that there are also people who carry a duplication of this chunk of DNA. And people that have a duplication of these genes show a reduced risk of schizophrenia. And this has been shown now in a couple of different separate genetic studies. So this is fascinating because it tells you that having too little of these genes increases your risk for schizophrenia, whereas on the other hand, having an extra copy of these genes than you or I reduces your risk. So it tells you that there's some kind of dose response relationship going on here. And you also know that at least one of these genes acts on the dopamine system. So this is the single largest genetic risk factor for schizophrenia, and one of the few protective, if you have the duplication, one of the few protective risk factors for schizophrenia. And there's good reasons to think it might act on the dopamine system. So we wanted to test whether it really did in vivo. We did a study in carriers of the 22 deletion and carriers of the 22q duplication who had never been psychotic, and we tried to scan them before the age of 25, so that they were still in this high risk period. And here are the data. So this is hot off the press. So again, on the vertical axis, you can see the level of striatal dopamine synthesis capacity, and then on the horizontal axis, you can see the parts of the striatum that we looked at, and the three different groups. So in black, you see the healthy controls. In blue, you see the deletion carriers. These are the people at high risk of going on to develop schizophrenia. Roughly 1 in 2 of them will develop schizophrenia in their lifetime. And you can see that they show elevated dopamine synthesis capacity compared to healthy controls. And then in red, you can see the dopamine synthesis capacity in the duplication carriers. And what's really interesting there is that they show lower dopamine synthesis capacity than the deletion carriers. And there seems to be a dose response relationship, which was significant across people's genetic loading for this risk factor for schizophrenia. The deletion carriers, the ones with the highest risk for schizophrenia, are those that show the highest dopamine synthesis capacity. So this is really interesting because it tells us actually that some of the genes that are risk factors for schizophrenia are acting on the dopamine system. And this is present even potentially several years, many years before they might develop the full-blown illness. So that tells us about genes, but what else might lead to the dopamine changes? What's going on in the brains of our patients that leads them to develop these dopamine changes and then psychosis? Well, generally it's thought that the dopamine elevation that we see in the striatum, shown here in this red area in the center of the brain, is actually secondary to changes going on elsewhere in the brains of our patients. And it's thought that it's loss of the cortical regulation of the subcortical dopamine system that leads to this, due to excitation inhibition imbalance in the cortex. This theory actually goes back to some early work by Rob Kerwin. For those of you who knew him and remember him, he did work in rodents that showed that lesioning the prefrontal cortex resulted in overactivity in the midbrain dopamine system projecting to the striatum. So this is the model that the cortical dysregulation is leading to that subcortical high dopamine that we see. So, as I mentioned, Rob Kerwin and others have shown that this occurs in rodents. What about in patients? Well, we've recently been looking at the relationship between striatal dopamine synthesis and what's going on in the frontal cortex, in particular in terms of gray matter volumes. And here you see the data showing that patients with schizophrenia who have smaller prefrontal cortical volumes are those that have higher or more marked dopamine synthesis capacity. So there is this relationship, as would be predicted from this model, between something going on in the frontal cortex and higher striatal dopamine synthesis. You'll notice there is something else that we see here, which is that we looked at patients who had responded with to antipsychotics, shown in black. We also looked at treatment-resistant patients, shown in the open circles. They don't show this relationship. Seems to be something else going on in them, and we'll come back to that towards the end of my talk. So this is interesting. It tells us that there's something going on in the frontal cortex that is linked to what's going on in the striatum. But what is it in this frontal cortex that leads to this? Well, the loss of grey matter volume in schizophrenia is generally thought to be due to the loss of synapses rather than nerve cells, because post-mortem studies have shown that people with schizophrenia, by and large, have similar numbers of neurons as you or I, but they do show a loss of synapses. So here you can see some photomicrographs from post-mortem studies looking at synaptic spines, and you can see quite easily here that the patient with schizophrenia shows lower synaptic spine density. Other studies have looked at other markers of synapses, markers that are present in the synaptic terminal, such as synaptophysin. And here you see a meta-analysis showing you that these also are reduced in schizophrenia. So there seems to be a loss of the connections between nerve cells that could lead to this loss of grey matter volume that we see in the illness. But this, of course, is all post-mortem. So one thing that we've been working on very hard in the last few years is trying to actually test that theory that there is a loss of synapses in vivo in patients. And that hasn't been possible until the last few years, because until very recently, it wasn't possible to measure any synaptic proteins in vivo. But a couple of years ago, a PET tracer that binds to a synaptic terminal protein was developed. And this is UCBJ PET. It binds to a synaptic protein called SV2A, which, as you can see, interacts with synaptophysin. And you get very nice PET images of the distribution of this protein across the brain, present in all synaptic terminals. So this is a marker that we can use to measure the density of synapses in vivo in patients. And we've now done that in schizophrenia. And what you can see on this slide is that on the vertical axis, the level of the binding of this PET tracer, so representing the level of synaptic terminal density, and healthy volunteers shown in orange, and compared to patients with schizophrenia shown in green. And this shows that there is a reduction or lower levels of this synaptic density marker across frontal cortical regions in schizophrenia, but interestingly not in the hippocampus, which is another area that we often think is involved in schizophrenia. And you can see on the right the statistical map of that, showing where these differences are. And again, the significant differences do seem to be particularly localized in the frontal cortex, in the grey matter, as you can see. So this is the first in vivo evidence that there is synaptic loss in schizophrenia. And it fits with this model that you've got a loss of synapses onto these glutamatergic neurons in the frontal cortex, and a loss of synapses onto the inhibitory GABAergic interneurons that regulate these glutamatergic neurons, so that you get this loss of or imbalance between excitation and inhibition, leading to overactivity in these glutamatergic neurons, some of which project down to the midbrain and are thought to then disinhibit subcortical dopamine to lead to the dopamine overactivity and psychosis. Meanwhile, in the cortex, of course, this loss of EI balance is thought to underlie the cognitive deficits and negative symptoms of schizophrenia, So this is a model that potentially explains both the psychosis and the cognitive and negative symptoms that we see in the illness. Now, I've shown you some evidence, that indirect evidence, that grey matter changes might be linked to subcortical dopamine disinhibition. But how can we actually test this? Well, we wanted to test this by developing a model of EI balance. And one model is to use ketamine. Ketamine blocks glutamate receptors, NMDA receptors, on these GABAergic inhibitory interneurons to reduce the inhibition of these glutamatergic projection neurons. And then the theory is to dysregulate the subcortical dopamine system. Now, that may sound all very academic to the clinicians amongst you, but actually this is something that you might come across in your clinical practice, because there are a lot of people that abuse special K. Now, by special K, don't worry, I'm not talking about your breakfast cereal, but actually, of course, ketamine. And ketamine is known on the street as special K, and is quite commonly abused. And interestingly, we know that acute ketamine induces both the positive symptoms that you see in schizophrenia, but also negative symptoms. And we did a study looking at chronic ketamine users in London who had then become abstinent, and found that they, even though they'd been abstinent for some time, showed ongoing schizophrenia-like symptoms, not at the level of reaching a diagnosis, but certainly there, and causing them some distress. So ketamine is certainly induces some of the symptoms that we see in schizophrenia, and repeated ketamine use seems to lead to persistence of these sorts of symptoms. So we developed a model based on this, a mouse model of repeated ketamine use. So the mice were given ketamine repeatedly over 5 days. They were then washed out of the ketamine, and then we did some scanning to see whether this might have reproduced as dopamine changes that you see in patients. So firstly, I just wanted to show you what effect taking ketamine has on the mice. So this is here, so some mice that have had ketamine and some that have just been given the control, and you'll be able to see that the mice at the bottom who had taken the repeated ketamine show this overactivity that's often thought of as an animal model of psychosis, although of course it's not psychosis. But it tells you that this model is having behavioral effects. What effect does it have on the dopamine system? Well, here are the data from that first study that we did, showing that even though they've been washed out of ketamine, those animals that had repeated ketamine showed a lasting elevation in their dopamine synthesis capacity relative to controls. So this tells us that EI imbalance, at least produced by ketamine, can lead to the same dopamine changes that we see in patients. What does this and the data that I've shown you in patients mean for treatment? In the last few minutes of my talk, I want to tell you about how we might develop this into new treatments that go beyond D2 blockers. So here I'm showing you in the middle a cartoon of the dopamine neuron and its synapse, with various sites at which we might act to treat the overactivity that we see in psychosis. And you can see on the top right that there is this postsynaptic D2 receptor antagonism, and shown with the little red symbol blocking the receptors. And that's, of course, what antipsychotics are doing by and large. But on the left-hand side, you can see the various potential sites at which we might act to downregulate dopamine activity. We're exploring several of these, but one I just wanted to show you is this traceamine receptor. This is present intracellularly in dopamine neurons, where it acts to reduce the firing of these neurons and also has some other effects to downregulate their activity. Now, why is this interesting? Well, several companies have now developed very selective drugs that activate this receptor and so might downregulate the activity of dopamine neurons. One of them is SEP856, and we've tested this in our ketamine model in collaboration with the company that's developed it. And here are the data. So in black and gray, you see the two control groups showing their dopamine synthesis capacity. In red, you see the animals that received repeated ketamine, showing again an elevation in dopamine synthesis capacity. And then in pink, animals that got the ketamine, but also got this TAR1 agonist. And you can see that that TAR1 agonist has restored dopamine capacity back to normal levels. So it seems that activating the TAR1 receptor has the potential to normalize dopamine synthesis capacity. So this is really exciting because this is evidence that for the first time that we might have a drug that acts on the dopamine neurobiology as opposed to acting post-synaptically. Of course, the clinicians amongst you will be saying, well, that's all very well, it's in animals. What might happen in patients? Well, the company have gone on to do a phase 2 trial and recently published this. And here you can see the data. So this is a study that compared placebo-treated patients with schizophrenia to patients that received this TAR1 agonist, shown in green. And you can see a significant benefit for those patients compared to placebo over the course of this 4-week trial. So this is a first clinical proof of concept, and it does seem that the compound is potentially effective. I think this is really exciting, but of course it needs to be tested further in further clinical trials, and the company have gone on to start a phase 3 trial, which is underway now. COVID has, I know, affected some of their recruitment, but they are progressing it pretty well. So we should hear about this in the not too distant future. And if that's positive, then this is something that may well come to clinics in the next couple of years. So I think that's very promising, offering a new potential strategy for treating psychosis. I want to finally just raise a few outstanding questions that we need to consider. And one of them that I've hinted at already is treatment resistance. Is there something different in these patients? They don't respond to D2 blockers. Another outstanding question, of course, from our in vivo findings on synapses is that's in patients who've already got the illness, but is it driving the onset of illness? So does that synaptic loss occur earlier? Now that is something we're currently working on, but I want to share with you in the last couple of minutes a bit of data on treatment resistance, because I think it's really interesting, potentially for clinical practice, but also for future directions. So at the moment, as I've talked about already, all our drugs, with the possible exception of clozapine, are D2 blockers, and we essentially are giving our patients with psychosis the same approach to try and treat them. So our treatments take a one-size-fits-all approach, but as we all know, patients come in very different sizes and different shapes of illness. And it's quite clear that there's a fair proportion of patients, about a third of patients, that don't respond to D2 blockers. a few years ago, we put forward a hypothesis that we should think of schizophrenia as at least having two subtypes. This was actually published in the British Journal of Psychiatry, and you can see at the top that British Journal of Psychiatry like to put pictures in and alongside their articles, and I was very grateful that they chose this picture of me when I was looking young and not stressed by COVID. But what we said in this article essentially is that there was a hyperdopaminergic subtype of schizophrenia, the type A schizophrenia, if you like, where they show this elevation in dopamine synthesis capacity and release. And these are the patients that respond well to D2 blockers. But there's another subtype where there isn't a major abnormality in the dopamine system. And these are the ones that don't respond to D2 blockers and are what we call treatment resistant in clinical practice. And we put forward the idea that perhaps this was a glutamatergic subtype of the illness. Of course, we're talking about two subtypes. There may actually be more than two subtypes. There may be a type C and a type D as well that have non-dopamine and non-glutamate abnormalities. But I think you've got to start with the major groups to begin with. And so this was the hypothesis we put forward. We've gone on to test this. Firstly, we did a study in patients with established treatment resistance and compared them to patients who'd responded to antipsychotics and looked at their dopamine synthesis capacity. And here are the data from that study. So here you see that those patients in blue who'd responded to an antipsychotic actually met remission criteria. So they'd done really well on their antipsychotic, nevertheless still show elevated dopamine synthesis capacity. But in contrast, those patients who are treatment resistant do not show that elevation in dopamine synthesis capacity. Their dopamine synthesis capacity was very similar to that you see in controls. So this is in line with the idea that there is this non-dopaminergic form of psychosis that's linked to treatment resistance. This was, of course, in patients who had established schizophrenia, who'd been ill for many years. The next question was, well, is this a consequence of being ill for many years, or is this present right from the onset of illness? So we then did a study in first episode patients who had yet to go on to any antipsychotic, and then looked at, once they started their antipsychotic, how they responded to that treatment. So here you see their baseline dopamine synthesis capacity and the vertical axis. And you can see that those patients that then went on to respond to antipsychotics showed an elevation in dopamine synthesis capacity compared to controls, whereas those that didn't respond essentially showed dopamine synthesis capacity that was very similar to controls. So this backs up the data that we saw in chronic patients to show that there seems to be differences in the biology present even from the onset of illness. What's more, we've been looking at whether we could use this dopamine measure to predict treatment non-response and treatment resistance. And here you can see some data on the sensitivity and specificity of using this dopamine measure. In fact, we've looked at various ways of analyzing these scans to see which is the most sensitive. And you can see the black line shows the most sensitive approach, which is pretty good at identifying those patients who will have treatment-resistant illness from those that are going to respond to antipsychotics. So this is, initial evidence that we could potentially, in the future, be doing imaging to stratify our patients. But what we also did in this study is looked at some economic modeling, and we wanted to see whether this would be a cost-effective strategy. So at the moment, of course, the only treatment that we have licensed for treatment resistance is clozapine. And clozapine is a drug that is complicated and has lots of side effects. So we figured that probably for a clinician to want to start a patient on that right from the beginning of their illness, they would want to be pretty confident that that individual had treatment resistance and no other antipsychotic was going to really work for them. So we set the specificity high at 95%, which of course penalizes the sensitivity. That means you miss some of those treatment resistant patients. So when we set it at 95%, depending on which analytic approach we used, we could identify between 40 to 60% of those treatment resistant patients. So that does mean we're missing a fair proportion, but we figured that trade-off is probably justified because you really want to be sure that you're getting people with treatment resistance before you start them on clozapine. And despite the fact we're missing quite a few, we showed that in the economic modeling that this is still cost effective. This is saving thousands of pounds, and this is based just on getting these patients to clozapine within a few months, when in fact, of course, we know that typically in clinical practice these days it takes years. So the cost saving, if you were able to reduce the time to get onto clozapine from years to weeks, is even bigger. So this is something that of course even managers would be interested in, and tells us that there is the potential here not only to improve clinical outcomes for our patients by getting those treatment-resistant patients onto the one drug that might work for them early, but it also has the potential to save resources. Now, just in the last minute or two, you might be wondering, well, what is going on in those treatment-resistant patients? I've suggested that it might be glutamate abnormalities, and that's one of the major theories of schizophrenia, of course, that there are these glutamatergic abnormalities. Are these more marked in treatment-resistant patients? Well, we've tested this in a few studies now, and it does seem that those treatment-resistant patients show greater glutamate levels in the frontal cortex than you see in patients that respond to antipsychotics. They're consistent with this idea that they have a different biology. Does this fit with clozapine? Well, potentially, because we know that clozapine has many actions, but there is some evidence that it acts on the glutamate system. And in a study that we've just recently published, we looked at the effect of clozapine on glutamate levels and found that patients that did better on clozapine, that showed more improvement in their symptoms, shown on the vertical axis here, were those that showed bigger changes in their glutamate levels. So potentially, clozapine might be working by reducing glutamate levels. Finally, how else could we target glutamate? Well, one strategy that's commonly used in treatment resistance in clinical practice is to give patients mood stabilizers like sodium valproate. And some of these, including valproate, do act on the glutamate system. Now, there is some evidence from meta-analyses that valproate is effective in this respect. So here I've got a meta-analysis showing you all the studies of adding valproate to an antipsychotic for patients that have not responded to that single antipsychotic. And you see that overall there's a significant benefit. But the problem is that this seems to be driven by the low quality studies. So when the meta-analysis then subdivides it by the open trials compared to the randomized controlled trials, you see that there is a much larger effect in the open trials, and when you just look in the randomized controlled trials, you don't see a significant benefit for valproate. So what this tells us is that whilst valproate may be effective, there is a significant question mark about whether this is really the case, or whether it's driven by those open trials, those low quality trials. So valproate is widely used in clinical practice, but there is equipoise about its effectiveness. So Cochrane said on the basis of this, of doing this meta-analysis, there's a real need for a well-designed double-blind RCT. And I'm pleased to say that we've now been funded to do this. That's a study that's just about to start, called the Atlantis study. And for the clinicians amongst you, here's an opportunity to take part and to help us advance understanding and clinical practice, because we now are looking for referrals of patients who might want to take part and help us test whether adding valproate to an antipsychotic really is effective or not. And I'm pleased to say that Cambridge and Peterborough are both sites in this study, and Emilio is the Cambridge PI, and Franco Orsucci is the Norfolk PI, and we also have Thea Bailey, who is one of the local researchers on this study. You can see on this slide that there are the various other researchers involved in the study as well. But this is an opportunity to get involved and help us advance the treatment options for patients with schizophrenia. And you can see the email addresses here, so please do refer and do contact your local PIs if you might be interested in contributing. So thank you very much for your attention. Here is the summary of what I've told you about today. Thank you very much.
Thank you very much, Oliver. A wonderful talk, wide range, and from animal models to very clinical practice. A round of applause. Thank you very much, Oliver. It was a fantastic seminar, very practical, very useful, and thank you very much.
But also do remember, everyone, think about the Atlantis study, and if you can contribute, please do get in touch with Emilio or his colleagues, and it'd be great to try and advance clinical practice by testing valproate. Thank you all.